Let's travel to the lower Amazon, near the city of Santarém, where the Tapajos and Amazon rivers meet to study the gentle giant Arapaima, which are among the world's largest freshwater fish, growing up to an incredible 3 meters in length and weighing over 400 pounds. They can be considered a flagship species for the conservation of Amazonian ecosystems because their protection requires the conservation of both freshwater and riparian habitats that have high levels of biodiversity. Their large size and succulent meat makes them a highly prized and valuable food fish. For some of 200 years, they have been among the most sought after fishes in the Amazon, but are now listed as endangered under the Convention on the International Trade of Endangered Species. And the International Union for the Conservation of Nature considers Arapaima to be data deficient, reflecting our poor knowledge of their conservation status and basic biology. Recent estimates suggest that Arapaima populations may only be 12% of their past abundances in the Amazon basin, reflecting widespread overfishing. In a proposed study area of Santarém, abundances may be at 4% of their past. In response to depleted Arapaima populations, local artisanal fishing communities are setting up community-based management systems where they assess population status and set appropriate fishing quotas. This has successfully rehabilitated Arapaima populations in a few areas of the Amazon. The sustainability of such efforts, however, hinges on our understanding of the role that Arapaima play in these ecosystems. For example, the capacity of a particular lake to support Arapaima depends on their feeding behavior and the composition of prey fish assemblages. In short, the conservation and management efforts for these iconic giants can be furthered by the study of their basic trophic ecology. I propose to study the ecosystem role of Arapaima using an integrative approach. I will study Arapaima diets by sampling their stomach contents, and I'll use stable isotopes to analyze their food webs. I will apply bioenergetic principles to understand consumption rates, and use this information to assess the biomass of prey fish needed to sustain healthy wild Arapaima populations. To study the effect of Arapaima on other fishes, I will sample fish communities along a gradient of low to high Arapaima densities. The aggregate of these studies will facilitate effective conservation and management of Arapaima populations near Santarém. Hello, my name is Capio Mandraker, and I'm a Ph.D. student at the State University of New York College of Environmental Science and Forestry. Your contributions, with the support of our research collaborators, both in Brazil and the United States, will help successfully complete this project. I thank you in advance for your generous support, and I look forward to keeping you updated. Your donations will go towards the following expenses: